a very interesting topic on data-driven perspective on drug discovery. But let me first give you some house rules. So when you join, so all microphones are muted and only the host can unmute them. But we strongly encourage you to ask questions by typing them in the question and answer window on your right-hand side. So periodically, what I'll do is I'll take your questions and I'll ask the speaker to respond to them. Some questions I'll wait until the end. And at the end of the talks, the speakers will stay back to take more questions. So this, this series has been going on for some time now. And there are, of course, after this lecture, there'll be two more. The next one is on, flattening the curve of the COVID-19 infodemic. It'll be on, the social media analysis of the COVID-19 epidemic. And after that, on May 18th, we will have a panel responding to global crisis in the age of data and information overload. So we really hope you'll also come back for those two lectures. So today, we have a very interesting take on, this is a topic where AI and machine learning can really shine. It's a data-driven perspective on drug discovery, and it'll be given by Dr. Ehsan Ullah, who's a software engineer in QCRI, and Dr. Raghvendra, who's a research scientist at QCRI. So each speaker will present for around 30 minutes. And as I mentioned earlier, some questions will be taken during presentations, but there'll be more questions, hopefully, at the end of the session. So I'll hand it over to Ehsan now to start. Thank you, Ehsan.
Thank you very much. Let me share my screen. Okay, can you see my screen?
Yes.
Okay, good morning, everyone. This lecture series is focused on ongoing pandemic of COVID-19. So we will be talking specifically about SARS coronavirus 2. Our group works in the area of bioinformatics and computational biology. Therefore, you would be hearing about computational aspects of the process involving drugs, their discovery, and their repurposing. Since we are computational scientists, we will not discuss virology in depth. This work is done by me, my friends Raghvendra, Abdurrahman Al-Basir, and Husam Al-Mir. The current session is divided into two halves. The first half is covered by me, and the second half is covered by Raghvendra. In the first half, I will start with the introduction to SARS coronavirus 2, the drugs repurposing, and how AI computational approaches are applied in this domain. In the second half, Raghvendra will talk about the process of application of AI for drug repurposing, and he will be sharing some interesting results that we have found through our work. So first, let's begin with viruses. The evidence of existence of viruses was first provided in late 19th century. One of the most interesting features of viruses is that they lie on the boundary of living and non-living organisms. They are very different from normal living organisms because of the following things. A new cell is directly formed in case of normal cells, and the normal cell divides into two similar cells through the process of mitosis, whereas a virus never directly forms from a pre-existing virus because it lacks the mechanism of reproduction. Therefore, the virus depends on a host organism through which it can use resources for its replication. And also, therefore, viruses have to modify the intercellular environment of its host. So talking specifically about the coronavirus, the SARS coronavirus 2 is an infectious agent that develops COVID-19 disease if it grows unchecked. It belongs to the coronavirus family of viruses, and this family is known to have a larger genome. Viruses causing MERS and SARS belong to the same coronavirus family. These viruses, SARS coronavirus 2 is likely to have come from animal sources, and with time it is expected to mutate. So far, there has been 3 subtypes that have been identified. Advancement of technology has made this the first pandemic in human history in which genome sequencing was rapidly completed, and hundreds of high-quality samples of this virus now exist. So this means that now theoretically we have the entire blueprint of the virus, and we can use this blueprint to perform different activities and understand its mechanism, such as how different proteins are produced from this virus, how it invades human cells, and how it replicates. Although the mechanisms are the biocascades that result from this information are not so self-evident, and a lot of experimentation is still required to understand all these mechanisms and understand the information about this novel virus. From the figure which is shown on the screen, the structure, this is structure of the virus. So if we look into the structure of the virus, it has an RNA strand, a single RNA strand, and which is covered by membrane proteins, and also lipid membrane. One thing which needs to be remembered here is that it has a special protein on its surface, which is called the spike protein, through which it actually infects human beings. And now let's see that how it infects the human beings. The coronavirus primarily infects the upper respiratory tract of human beings and mammals as well. The MERS coronavirus has been shown to bind to a protein DPP4, which is expressed in non-ciliated cells in human beings. In SARS coronavirus 1, the ACE2 protein was the primary cellular receptor to which the virus attached itself, and it is believed to play a really important role in the ability of SARS coronavirus 1 to produce infections in both upper respiratory tract and the lower respiratory tract. Another important factor that contributes to the state of SARS coronavirus 2 patients is the immunopathogenesis. The immunopathogenesis is development of a disease as affected by the immune system. So it is the immune system of the patient which is actually activated, and because of that response, it releases inflammatory markers which initiate a positive feedback loop to form a condition called acute respiratory distress syndrome. This syndrome actually fills up fluids that you can see in the small air sacs of lungs, and which causes the ability of reduces the ability of the lung to absorb oxygen from the air. So which causes shortening of breath in SARS coronavirus 2 in severe infections where the immune system of the patient has been activated. Also, one thing to be remembered here is that it is known that this virus has come from bats. And one thing which is important about bats is that the immune system of the bats doesn't respond to the virus as the immune system of human beings respond to this virus. So that's why the bats don't get much sicker from this virus, and they are good carriers for this virus. So now let's go through the way that how this virus infects human beings at a cellular level. So here I am showing you a cell which is shown in the lower part of the slide, and the upper part shows the extracellular environment. So in the extracellular environment, you can see macrophages, which are the cells from for the immune system, and also some other kind of receptors. And this is the boundary of the cell. And here on this boundary, you can see there are different small things, and which are the receptors available on the boundary of the cell. These receptors are actually responsible for active transportation of different things across the cell membrane. So what happens is that when a SARS coronavirus tries to infect a cell, it attaches itself to one of the receptors, which is ACE2 receptor, through its S protein or the spike protein. And by the interaction of this spike protein to TMPRSS receptor available on the surface, this enables the membrane fusion and endocytosis, and the virus enters into the cell. So once the virus enters the cell, it uncoats its envelope, and the RNA of the virus is released into the cell. From this released RNA, the translation happens, and polypeptides are generated, and non-structural proteins are manufactured, which later are used to produce RNA of the virus and structural proteins, and new viruses are assembled, and those viruses are then excreted to the extracellular environment through the membrane. So now let's talk about the drugs. The drugs are for combating the viruses. The primary defense line is the vaccination. The vaccinations create artificial immunity, and it would be the quickest way to stop the pandemic. And without the vaccinations, the next defense line is use of antivirals or other drugs. In case of SARS coronavirus 2, since this is a novel virus, therefore there is no vaccination which exists so far. So at this point, the antivirals are the primary line of defense. The antivirals are a class of small molecules which inhibit activity of a virus by disabling some part of the virus. One of the good examples of an antiviral drug is HIV. And since there is no vaccine for HIV so far, the main line of defense are these antivirals. As a rule, just remember, the antivirals work by targeting and binding chemically with virus proteins to disrupt the life cycle of the viruses, so that the reproduction of viruses can be stopped and the infection can be controlled. So far, practically speaking, there are no approved drugs for SARS coronavirus 2 because of its novel nature. Therefore, the mortality and there is no drug available which can control the mortality because of infection of this virus. So let me walk through the history of drug discovery so that we can develop an idea that how our drugs were discovered, how in modern days the drugs are discovered, and how this drug discovery can be used for repurposing of drugs, and how computational approaches can be used in this domain. So historically, drugs were discovered by identifying the active chemicals from traditional remedies. The discovery of quinine is known to be one of the most serendipitous medical discovery of 17th century to treat malaria. And quinine marked the first successful use of a chemical compound to treat an infectious disease. With advancement of pharmacology, active substances could be extracted from the crude material, and later these were synthesized synthetically in the laboratories. And in this case, the biological target is not exactly known. Rather, the more focus is on the biological activity being done by these these compounds, which means that if a drug is controlling a biological activity, which is, for example, spreading of infection, this means this drug is actually working. Whereas in case of modern discovery, the small molecules are discovered to target specific proteins maybe. So I will be using the term targets for these specific entities to which the drug is going to bind, and it will be having an impact to control the conditions. And besides that, there are certain other factors which must be considered for the drugs, which are the toxicity and its efficacy. The small molecules which can, there may exist many small molecules which can address or which can attach themselves to certain targets and control the infection, but they may be toxic and may increase the mortality rate if used. So we need to consider all these side conditions as well. So in this slide, I'm showing you the process of drug discovery, which goes from left-hand side towards the right-hand side. The first half is the drug discovery, and the second half is the drug development. In case of drug discovery, the first part is the strategic research, in which a target is discovered. So by target, again, we mean different proteins which can be targeted to disrupt the life cycle of the virus in case of development of an antiviral drug. So once the target has been identified, the exploratory part of the research starts, in which small molecules are screened and selected which can be potential drugs, and those small molecules are selected such that they can have a high binding affinity to these targets. And once a candidate set of these small molecules has been identified, they are from them the lead molecules are identified, and then they move on to the next stage, which is the candidate drug selections. As I have told you that not all molecules which show a high binding affinity to the targets are useful, because they need to be tested. So the primary thing for testing these molecules is ADME prediction. ADME stands for absorption, uh, their metabolism, their excretion, and then once the molecules which have which have passed this stage are used in the animal models to check in vivo studies, and once the molecules which have been tested in the animal models become the candidate drugs, and then they move to the next stage, in which the clinical trials and approvals from different drug authorities is performed, and then they move to the market. So in case of SARS coronavirus.
Sorry, Ehsan, a quick question. So where does that in that life cycle, the pipeline you were showing, where is the role that AI, in which stage can AI and machine learning play the most important role? Would it be in the
Yes, that's an excellent question. In this particular pipeline, one of the major things is that AI can play a real important role in exploratory research part, because this is the part where we can go through the libraries of different chemical compounds which are available, and since the targets are already known, we can perform computational approaches to see that which compounds can actually bind to these targets, and then select the lead compounds which can go to the next stage of candidate drug selection. So the AI can actually help, and this is what we have our work focuses on, is the exploratory research phase. Is that clear?
Yes, thanks.
But yeah, and by the way, one important factor here to remember is that this whole pipeline takes a lot of time, and on average, the cost to bring a single molecule or a single entity to the market requires at least $2 to $3 billion. And the total time required for this whole pipeline is approximately 15 years. Okay. So the next thing is, now let's see that for coronavirus 2, which are our primary targets, and then we will see that what are the possible drugs which can be used here. So as I have explained you earlier, that uh, on the boundary of the cell, which is the cell membrane, there are different receptors. So if somehow we can have some inhibitors which can attach to these receptors can be blocked. And once these receptors are blocked, the SARS coronavirus cannot attach itself to these receptors and enter the cell. So one potential target here can be these receptors on the cell membrane to avoid SARS coronavirus infection. Another possible target can be the proteins which are allowing virus to enter the cell. The next possible thing is the inhibitors for proteolysis, which are converting the polypeptides into non-structured proteins. And the next potential target can be to inhibit the mechanism of production of RNA of virus for generation of new viruses. So in this whole process, there are some educated guesses. Like, for example, here to inhibit membrane fusion and endocytosis, arbidol and chloroquine, hydrochloroquine can be used. Lopinavir can be used to inhibit proteolysis, and ribavirin, remdesivir, favipiravir. These are the medicines that can stop production of viral RNA. So these are some kind of educated guesses which I am showing you here. And now let's see what is state of the art right now. In the media, you might be seeing that there are in the news many drugs are being discussed. Like, for example, many anti-malarial drugs have been shown to be effective, and there are studies and ongoing trials showing different antiviral drugs to treat this particular virus. Based on NIH's website, there are about 350 active trials going on, and only minority of these are related to antiviral drugs. So the process of conducting a clinical trial is long and costly, but it is absolutely scientifically and legally necessary to get a medication approved because of its lethal effects. So now, given that although we have there exist databases of thousands of chemicals, and for testing them, we can have a lot of coronavirus patients. Even a lot of resources are required to conduct these trials. And also, it is almost impossible to test all the potential candidates as well. And also, like, some of the chemicals like chloroquine are known to have some side effects. So there should be some kind of pre-screening which can help in identifying that which potential chemical compounds can be used for treating this virus. And remdesivir is a controversial because even though it is just approved, it has not shown a clear decrease in the rate of mortality. Just remember that the evidence so far is that this drug is used to control the symptoms, not to control the mortality. And the state of the art in current pandemic situation now is either to develop a vaccine for developing the artificial immunity, or use an existing antiviral to avoid this long process of discovering a new drug to treat this infection. So the use of an existing drug to treat an other medical condition outside the scope of the original medical indication is called drug repurposing. So given a large number of already approved drugs, there is a significant chance that some of these could help to treat COVID-19 disease. There are many advantages for the drug repurposing, especially in the current situation. For example, since the repurposed drug has already been found to be sufficiently safe in pre-clinical models in humans, it is less likely to fail, at least from a safety point of view. And the time frame for the drug development can also be reduced. As for the new drug, there are substantial savings as well. Because it has the concurrent drugs have been passed through a lot of animal models testing and for their efficacy, and also less investment is required because less resources are and lab time is required. And moreover, one of the advantage of repurposing drugs is that they can reveal new targets and pathways that can be further exploited. And these advantages have a potential to result in less risky, more rapid return on investment in the development of repurposed drugs. And also, the average cost of development can be reduced. And just to let you know that on average, it is estimated that for repurposing of a drug, approximately $300 million are required, which compared to $2 to $3 billion for development of a new drug. And the time frame you can see can also be reduced compared to the drug development, which was around 15 years on average.
So Ehsan, have been also that's my question actually. Are there successful examples? So that's what you're showing. Great, thank you.
Yeah. So here are some of the successful examples of drug repurposing. Drug repurposing has been used in the past. Like for example, Zivuduvine was originally used for cancer in 1960s, but in 1987 it has been used to treat HIV/AIDS. And this happened due to different repurposing methods like in vitro screening of compounds. So in this slide, I'm showing you different examples where different repurposing methods have been used. Another drug, minoxidil, was used for hypertension in 1963, and it was discovered as a side effect that it can be used to treat hair loss. And in 1988, it was actually marketed for a hair loss control medicine. Another very well-known medicine which has been used for repurposing is sildenafil, having a brand name of Viagra. Practically, it was developed for angina to treat angina conditions in 1998. But interestingly, during its clinical trials, it had many side effects. And one of the things was discovered that it was very effective for treating erectile dysfunction. So during clinical trial, it was repurposed for erectile dysfunction, and it was marketed for this particular condition. Similarly, other medicines like fingolimod, which was used for transplant rejection, is used to treat for MS. And this is the first medicine which is being used to treat multiple sclerosis. Another medicine which is very well known, aspirin, was actually developed in 1897, and now in 2015 it has been used to treat colorectal cancer. And in the very right column, you have seen that there are many repurposing methods which may involve screening of the compounds in the lab, or maybe there were some side effects of a drug which was being exploited, or maybe retrospective clinical analysis can be used, or some other pharmacological and clinical analysis can be used to identify other useful purposes of the existing drugs. So if I'm going to discuss these drug repurposing approaches, there are primarily two major groups or categories for these drug repurposing approaches. When here I'm talking about drug repurposing approaches, I'm talking about the second stage in the drug discovery pipeline, which was exploratory research. Okay, so here in this case, when I'm talking about drug repurposing approaches, these approaches correspond to the exploratory research phase of the drug repurposing. So in this case, the identification of the right drug for an indication of interest with a high level of confidence is very critical. And it is this where modern approaches for hypothesis generation could be most useful. These approaches are divided into two prime categories. The first is experimental approaches, which are highlighted in green in the pie chart shown on the right. And the other approaches are the computational approaches, which are highlighted in the blue. The phenotypic screening can identify, in the experimental approaches, the phenotypic screening can identify compounds that show disease-relevant effects in the model system without prior knowledge of the target. So again, it follows the classical approach of identifying the drugs. Whereas the binding assays can identify interactions in terms of binding of a drug to the desired target, which is the modern approach where the target of any disease is well known. And talking about the computational approaches, which are highlighted in blue, we will be focusing mainly on novel data sources, genetic association, signature matching, and molecular docking, whereas retrospective clinical analysis and path mapping I will not be focusing on. And I will give you the reasons why we are not focusing on that. But just to remember that these approaches will be used to develop models that can use prior information to compute whether a drug will inhibit a target or no. So here are the approaches which I will be focusing on, as I have already told you, are novel data sources, genetic association, signature matching, and molecular docking. The two approaches which we will not be focusing on are retrospective clinical analysis and pathway mapping, because we don't the retrospective clinical analysis requires electronic health record data from electronic health records, and we don't have access to them. And regarding pathway mapping, our group doesn't have much expertise in this area. So one thing which I would like to mention here is that for rest of the approaches which I've shown on this slide, our group have developed expertise in these techniques to develop models to predict sensitivity of drugs to treat cancer, which is a real modern area of personalized medicines. And moreover, since a lot of data is available publicly in form of different databases, so we can exploit that data to identify drugs that can be repurposed. The first major area of drug repurposing approaches is availability of novel data sources. Here I have listed a few of them. PubChem contains over 25 million compounds and 90 million compound activities. Drug Bank has over 2,500 approved drug information, including the targets, which can be proteins, enzymes, transporters, and mechanism of actions as well. UniProt has over 13,000 viral proteins and 2 million other viral proteins which are not manually annotated. So this particular database has both annotated and unannotated information about viral proteins. And regarding the drug activity, we have Campbell, which contains over 50,000 curated drug activities, PubChem, BindingDB, which contains binding affinity scores for drugs and compounds to different proteins. And also there are some other websites or databases, for example, Drugs Virus Info, which contains information about drugs which are used for different clinical trials on variety of viruses. And it shows the progress of these clinical trials for drug repurposing. And Chemical Checker, it contains information about drugs which are similar to other drugs based on the literature and chemical techniques. And this is also a really important source for drug repurposing. The next approach, which can be a computational approach, which can be used for drug repurposing, is genome-wide association studies. Genome-wide association studies actually identify potential targets at genome level. And this provides insight into disease biology and help identify novel targets. These targets might be shared between diseases and thus help in drug repositioning as well. But there are certain challenges associated with GWAS. For example, in gene-rich loci, where strong linkage disequilibrium is present, identification of causal genes becomes really very difficult. And also these studies doesn't provide information about direction of effect of the gene variant, which requires further functional analysis to check if the variant has has an inhibiting or the promoting effect. And also GWAS requires further downstream analysis to identify proteins and pathways which can be potential targets for drug repurposing. The next approach which is used for computational drug repurposing is signature matching. Signature matching is based on comparison of unique characteristics or signature of different entities, such as drugs, diseases, and clinical phenotypes. These signatures can be obtained from different types of data. For example, transcriptomic data, proteomic data, metabolomic data, and chemical structures of the drugs. The transcriptomic structure is derived from gene expression profiles obtained from tissues. And these tissues can be from the controls. These can be from the cases, which means from the tissues have been infected with the viruses or with the disease. And these tissues can have also be from without treatment, and the signatures can be obtained after treating the tissue with the specified drug. The metabolic signature can be derived from the concentrations of different metabolites in those tissues. And similarly, the proteomic signatures are different protein sequences available for different viruses and inside the tissue. And the chemical signatures are derived from the chemical structures of the different molecules. And one important thing to remember is that these structures can be represented as molecular images, or these have simple representations such as smiles, which is a linear representation of the structure of the protein in form of a string consisting of different characters. Now, by matching signatures, we can have like, for example, drug-drug similarity and drug-drug similarity can be identified. The drug-drug similarity can identify further similarity between the mechanism of action of drugs, suggesting that given a drug can be used for a therapeutic purposes, another drug having a similar structure can be a potential candidate to be used for the same therapeutic use. If I'm going to talk about drug-disease similarity, the drug-disease similarity can identify which drugs can be used to treat a disease. And again, these approaches have their limitation. For example, there can be errors in chemical structures, and also different drugs have their unique side effects. But by incorporating the side effect information, we can compensate for the side effect thing as well. And the last approach for that can be used for computational drug repurposing is computational molecular docking. This approach actually predicts the binding, the site complementarity between the drug and the target. This approach is a structure-based approach, and it can identify interactions between drugs and the targets. And there are again limitations to such approaches as well. Like it requires 3D structures of protein targets, and if the protein structure is not available, the docking cannot be performed or cannot be determined. Also, there are not that many databases which contain accurate structural information of targets. And finally, the quality of algorithms is a question, has been in questions, because there is still a lot of room to improve, for improvement, and confidence in predicted binding affinity scores is really low. One of the solutions based on these computational approaches, as described earlier, has been presented by Barbasi, and they're using a network-based approach. This slide shows physical interactions as used as input of the framework. So on the left-hand side of this slide here, you can see viral human protein interactions. These are capturing human proteins to which viral proteins can bind. The middle part here shows human protein-protein interactions, which define the human interactome linked pairwise and the physical interactions. And on the right-hand side, human drug protein interactions are shown, and these capture human protein targets for each drug. Now I will hand over the presentation to my dear friend Raghvendra, who will explain that how we have used these approaches to identify different drugs that can be repurposed for treatment of COVID-19. And before that, Sanjay, if you have any questions.
Yeah, so if you have questions, just please put them in the Q&A box. Otherwise, Raghvendra, you can take over while,
Yeah. Okay. Can you see my slides?
Yeah, we can see them.
Okay.
You can make them in, yeah.
Okay. So before we talk a little bit about the models and some of the results that we have, what I would like to say is that we focused on making, sorry, yeah, I'll just switch on my camera. Yeah, we focused on some of the information sources which are available, and we tried to formulate the problem in a simplistic way, but at the same time we show the power of how machine learning and AI tools can be used given the formulation that we are using. So what we are focusing on is the drug, given the drug and a virus, and the virus, which in this case is the SARS coronavirus. We want to see how effective is this drug against the virus. So many of the things that I am saying in here are surrogates. So for example, drug is a surrogate for a ligand. This equation is actually coming from the ligand receptor binding affinity, where the drug is nothing but a ligand. The virus is nothing but the receptor, where basically you have viral proteins that Ehsan explained, that there are many of these different viral proteins. And what we want is that the drug basically binds to these viral proteins and prevents its functionality. And to do that, how this is done, or how the activity is estimated, is that you take different concentrations of the drug, and then what you want to see is that at low concentration, the drug binds to the viral protein and inhibits its function. So what is done in the laboratory is that at different concentrations, you measure the activity and you try to extrapolate and get the full area under the dose response curve, which gives you a complete picture of how the drug is effective against the virus. And several of these quantitative measures, which are based on the half maximal response of the drug against a receptor, in this case it is the viral protein, are present in many of these databases that Ehsan mentioned, particularly in PubChem and Chembl. And these quantitative measures are called like IC50, EC50, drug efficacy, and some of them which Ehsan already named. So what we are looking for is that what is that concentration at which 50% of the viral protein is inhibited by the drug, which is referred as IC50 in literature. And this can be used easily. This particular framework can be used for machine learning techniques. So machine learning techniques allow you to model this, and how do they allow you to do this will be explained in the next few slides. So our problem formulation looks like we want to estimate a function given a drug and a virus. We want to learn the activity. And why will this work? The reason it works is because you have some information in the drug. So these drugs are made up of chemical atoms, and the virus has these viral proteins, which are again made up of molecules which interact with one another. So what we are looking for is basically co-occurring patterns that increase, basically that can tell you that how good is the activity when these co-occurring patterns in the drug and the virus appears. For example, you can see that there might be presence of a carbonyl group and multiple histidines in the viral protein, which drives the activity or which allows the binding to happen. So where is this information encoded? This information can be encoded in the drug's representation as well as the viral protein representation. So the viral, in the case of the drugs, you have the representation of the drug in the form of a structure as an image, as you can see here, or you can use the SMILES representation, which again encodes all this chemical information in a linear sequence. The same goes for the viral protein. In the viral protein, it is made up of a set of 20 amino acids, and in its primary structure, it is just a linear sequence of these amino acids which are present. So there can be 20 such possibilities, and it forms a sequence. However, there are more information available about the drugs which can be used. For example, for each of these drugs, you also have structural image, structure which is in the form of an image that can be used for machine learning model. You have also mechanism of action available for many of these drugs, as Ehsan mentioned, in the drug bank, which is generally in a summary form. So you have basically a paragraph which can be considered as a text. And you also have physiochemical properties, which are basically numeric features like molecular weight, log p value, solubility, and features associated with the drug. The same goes for the viral proteins. For the viral proteins, you have a primary sequence of the amino acid, but you can also have 3D structure or images of various viral proteins produced by a particular virus, including and you can also have functional annotations, which can be basically what kind of biological processes are these viral proteins involved in. And lastly, you can also have physiochemical features such as, again, molecular weight, aliphatic index, gravity index. There are many of these characteristics which you can have for these viral proteins. Why we are focusing on the amino acid sequence or the primary structure is because that not all of the viral proteins that are being produced by the coronavirus, we have high quality 3D structure of the viral protein. So that is one of the reasons that we focus on the primary structure. And the second reason is that all the information that is required when we are looking to capture the co-occurrence is already present in your primary structure. So basically the presence of histidines or the presence of carbonyl group are already encoded in the SMILES as well as the linear amino acid sequence representation of the drug and the virus. So given this information about the drug and the virus, now and you have labeled information about activity of these combination of drug viral proteins from resources such as PubChem and Chembl, can we model it? And how can we model it? So we can model it using traditional techniques like...
There was a question, I think is the right time to answer.
Yeah.
It was this, can you talk about the role that biomarker discovery plays in the process? Will you come to that or?
So in this case, I mean, in our simplistic model, we are not taking into account the mechanism of action. The representation that we are looking is only the sequence representation of the SMILES as well as the representation of the sequence representation of the protein. So we are not taking into account if the question, if the questionnaire is asking about what is the mechanism of action, like how the drug interacts with various human protein or with the viral protein, we are not taking this into account, like the mechanism of action. And so it's hard to discover the biomarker as such. What we are looking for is that the presence of something in the drug as well as the presence of something in the viral protein, this together suggests that there is a high binding affinity or there is high activity to inhibit the virus. So we are not necessarily looking at biomarkers in here.
There was another question which could be related, like how are you choosing the drug candidates? Basically, how are you choosing your databases? Because there are millions of drugs available, right? So how do you select?
So that's a very good question. I mean, it's answered in the next slide, but yes. So what we are choosing is basically all those drugs which have been used against viruses which are either in the family. So initially we started with the viruses which are similar or belong to the family of coronaviruses. Like we took the MERS coronavirus, we took the SARS-CoV-1, we took so all of these coronaviruses, and from the databases like PubChem and Chembl, we focused only on those first. But what we saw is that the dataset size is very small in that case. The set of drugs that we were looking for, we wanted to use all possible drugs which have been tested against any of these viruses which are similar to the coronavirus. But then we expanded our pool by taking into account also viruses like or viral proteins belonging to the HIV family, the Hep C family, because we are seeing anecdotal evidence of drugs being used for HIV having some impact on the SARS coronavirus also. So we expanded our search space by including all possible viral proteins, and the other thing that we also did is that we only focused on those. So here I showed 3 viral proteins, right? And throughout our work, we will focus on only these 3 viral proteins, because for 2 of them, the MERS and the SARS-CoV-1, both of them, many of these drugs have targeted these as the main protease for MERS as well as for SARS, the PL-PRO and the 3CL-PRO, which is the papillon-like protease and the cleavage protein. In the case of coronavirus, as Ehsan explained multiple times, that the spike protein is the one by means of which it enters the human cell. We also focused on the spike protein. But in literature and like in these databases, you have already information that most of the time people are targeting only the main proteases of these viruses. So for the SARS coronavirus, it produces 22 different proteins, but here we focus only on these 3 proteins. One, because of the high quality 3D structure that is available, and second, because what we have observed in literature is that for the MERS as well as for other coronaviruses in their family, people have targeted using drugs, the NSP3 as well as the 3CL-PRO. So this is the way we have defined our search space. Thanks a lot for this question, because it allows me to give you more details.
Great. Yeah, continue. Thank you.
Thank you. So coming to the machine learning methods. So now we have, I talked to you about that you have these, we are taking into account when we are building the models, only the SMILES representation for the drug, as well as the sequence representation for the protein, right? But many of these machine learning methods, they need vector representation of the data to do their function estimations or to do the activity prediction. So when I say vector representation, what it means is that they either need a numeric representation or a one-hot coded representation of the categories that are there in your data. So a simple way is like what I just said, you can convert your data into a categorical form, but what that would lead to is an explosion of your dimensions. And the other way is that you can come up with a numeric representation of the SMILES as well as the amino acid sequence. But by doing that, though you retain all the information, the chemical information which is there in terms of numbers, but you cannot really go back and do a mapping, one-to-one mapping to see which number corresponds to which particular, say, the SMILES character or the amino acid. So you lose a little bit in terms of representation. But just to explain a little that how these machine learning methods work. So in the case of random forest, once you have the vector representation of the data, you use a tree-based method to basically identify sets of features in this representation that can predict a particular activity value, and you fit many such trees and take an average of these prediction. It's basically a bagging based approach. Whereas in the case of XGBoost, what you do is that you fit a tree based on the feature representation that you have got for the drug as well as the virus, and then whatever error is remaining in the activity that you have estimated, you simultaneously or iteratively keep fitting on that with trees until your error become very low. And this approach is called boosting based approach, and this a method that we are using is called XGBoost. In the case of support vector machine, what you are trying to do is that if your true activity functions look like this green sinusoid curve, which is here, given the discrete set of drug virus combinations and their activity values, we try to estimate from these discrete set of points the true function which is there using a nonlinear and convex optimization based approach. So all of these approaches are nonlinear by nature, and they can capture the interaction between the drug components as well as the viral protein components while making their predictions about the activity value.
So Raghvendra, your approach seems to be more like, okay, we have these massive databases for drugs, for virus activity, and essentially, you know, it's a data integration problem, right? Yes, and you want to put all these different databases in a common language, a representation where you can essentially, so that then you can feed them into a machine learning classifier or something. So it's essentially about how do you connect these databases and then put them in a common representation language. Is that the right way of thinking about it, or?
I mean, 100%. So majority of our time is always spent on collecting and curating the data. So I said to you, it seems very easy that, you know, in PubChem and in Campbell, you have information about this. But when you try to constrain your, put in all, given all the constraints of your search criteria, when you try to curate and integrate the databases, that becomes the toughest part of this entire problem. And once you have got all the information, then you can use, I mean, the vector representation is just one form, but you can also use just directly the SMILES representation or the amino acid sequence. Doing the machine learning modeling becomes very easy, but collecting and integrating, as you said, and curating takes more than 80% of our time. And exactly, it took more than 80% of our time.
Thank you.
Yeah. So as I just mentioned, that you can, so either you use the vector representation, but the power of modern AI is that you can even use the sequence representation that you have for the SMILES, or I mean for the drugs, which is basically the chemical descriptor SMILES, as well as the sequence representation for the viral proteins that you have in the form of the linear chain of amino acids, and directly pass it to a deep learning model. So here I give you an example that I have to do, suppose an NLP, I mean a natural language task, like given a sentence which can be considered as a sequence of words. I want to output whether my sentiment is positive or negative about this. And what this particular deep learning model does is that it takes into consideration this sentence and converts it into a matrix of numbers. And then what the convolution filters are doing basically are looking at adjacent terms with, basically looking at local context which is present in this particular sentence, while trying to predict ultimately the sentiment which is involved. So the CNN based neural networks, what they allow is to capture the local context which is present in your input sequence, and the context that it learns are driven by the true prediction values, or are driven by the sentiments that we have. So this is the way in which we can also, we use this as an analogy when we are also trying to make our models. Similarly, you also have recurrent neural networks. What they allow is that given the sentence, they allow you to have memory gates. So basically you can have even long-term dependencies. So short-term context can be captured, but also you can capture long-term dependencies when you are trying to make ultimately your prediction. So why is the LSTM important in the case of, for example, our drug viral protein? It's important because, as I mentioned a little bit earlier, that for the viral proteins, you also have 3D structure. So it can happen that we are using the linear representation of the protein, but this protein can be convoluted and have 3D structure, and an amino acid which is closer to one terminal or the C terminal might be interacting with another amino acid which is closer to the end or the N terminal, and together they are providing a closed form or a conformation. So that kind of dependency can be captured much better by an LSTM based model, because it allows you to have the memory gate. So given that we have explained that for the traditional machine learning model, we need the vector representation of the drug and the vector representation of the protein sequence, whereas we can also make end-to-end deep learning model where we can just use directly the sequence representation of the drug and the sequence representation of the protein sequence. What we are also, and as I mentioned through the question that Sanjay asked, that by doing a data integration, we have more than 60,000 such samples of drug virus activity information from Campbell and PubChem. Our ultimate goal is to estimate this function using both these traditional machine learning methods as well as end-to-end deep learning. So to get the vector representation of the drug as well as protein sequence, what we initially do is we use an LSTM based autoencoder. So in the explanation that I gave about the sentiment analysis, I showed you that if you are given the sequence, you are trying to predict an output, right? In the case of an autoencoder, what you do is that you are given a sequence and your output is the same sequence. So basically what you're trying to learn is that what is the innate hidden representation which is required, which is applicable for this particular drug in low dimensional numeric representation. Can I get that low dimensional numeric representation while reconstructing the same input? In here, if my input is carbon dioxide and the output is also carbon dioxide, so I want, given this as input, I want to minimize this, but at the same time learn a representation which is valid for all possible drugs and is numeric, and in the sense that it can be used by my traditional machine learning model. So for doing that, we use an LSTM based autoencoder. So initially what we do here is that we use databases like the Moses and PubChem to get 2.5 million compounds. We screen for compounds having salt bridges and compounds whose SMILE structures are having more than 120 characters or are basically a large compound. We remove them out, and after filtering from them, we do this procedure of reconstruction. What we observe is that on average, You can see here that our training as well as validation losses are very low. On average, for a per sample, we have a reconstruction error of like 0.001, which means like there is almost no error in our reconstruction. What we give as input, we get as output. And a more stringent condition that you can use is that the compounds that you are giving here, the model does not know any semantics about whether the smile representation. So what is possible is that when you have got the smile representation, there are tools like RDKit which allow you to see whether this smile representation is for a valid compound or not. And though in the smile representation is nowhere present whether it is a valid compound or not, when we train our autoencoder and we get the reconstructed smile, what we want to see is that the reconstructed smile is also a valid compound. And for approximately 97% of the compounds that we trained on, we get a reconstructed compound which is valid. So it's a more stringent condition, but it is captured by our model.
So Raghvendra, if you have such a low reconstruction error, is there a danger that you're overfitting, or because your bottleneck, in autoencoder is all about the bottleneck. How big is your latent space, in some sense? So what is the explanation that your reconstruction error is so low?
So I mean, ideally, won't we want because it's an autoencoder, we want the reconstruction errors to be low as possible, even if it kind of overfit, because it's not being used directly in a supervised framework. It is unsupervised, right? And the more stringent condition or the more satisfactory condition is that the reconstructed compounds that you have are basically true valid compounds. And though we also divide the data to overcome the bottleneck of overfitting. We have divided the data into a training and a validation set, where the validation set is completely independent of the training set. So even on the validation set, we have low errors, which is a good characteristic of the model.
Thank you.
And a similar procedure is used for the protein sequence, but instead of the LSTM, we use a multi-layered CNN-based model to get the vector representation for the protein sequence. So this is, one of the reasons why we chose because we have in previous works, we have shown that whether we use a LSTM-based representation or a CNN-based representation, the reconstructions are more or less similar. The reconstruction error is also very small. Also, another constraint is that the length of the input sequence of the protein is, we fixed it at a maximum length of 2000. And the LSTM models are much slower when your input sequences are too long because they are sequential by nature. Whereas CNN models can parallelize majority of the computation. So that's one of the reasons. But now once we have the vector representation for the drug as well as for the virus, we can concatenate the two and use it as a feature representation of the drug virus representation. And we have for each of these combination of the drug virus, we know the activity value, which is the standardized p-Campbell value that we use in our experiments. So we can now pass it directly to the traditional machine learning model. But we can also make use of end-to-end deep learning models, where what you can do is that you give the smiles representation, and then using a CNN-based encoder, you can get a numeric representation for the drug, as well as you can do the same for the proteins. And once you have got this numeric representation, you can connect the two and concatenate the two and pass it to feed-forward layers, which are nothing but multiple hidden layers, to have nonlinear interaction between the drug representation and the protein representation, and ultimately predict the activity. So the difference here in comparison to the unsupervised methods is that all the representation that you are learning are being driven by the true activity prediction that you want here. So the losses or the errors that you make here backpropagate, and they drive the representations that you're learning for the drugs as well as for the proteins. So that is one of the key difference between the traditional machine learning. So you're learning more from a bigger data set in the case of the traditional machine learning models when you did the unsupervised part. But in case of the end-to-end deep learning model, you can only train on the 60,000 samples that we have, and we cannot explicitly use the additional information. We can, but some machine learning approaches do allow you that, but we have not tried in this particular framework. So again, here we can use, I showed you an end-to-end CNN-based model. Here we can also use an LSTM-based model, which can capture the long-term dependencies better. And we can also use a CNN plus LSTM model. So the output of the CNN are then passed to the LSTM, which will capture, instead of capturing like long-term dependencies from the primary sequence, it will capture it from the nonlinear representations that the CNN has learned. It will capture long-term dependencies between them. So we can use all of these different type of models. So now I will show you some results. So for doing all of this, what we did is that we divided our data into a training set, and we used some part of our data as a test set, where none of this test set is available to our model during training. So during training, we do 5-fold cross validations and like hyperparameter tuning, but none of the test set is visible to the training set. So, and the number of, one important thing that I've not mentioned, but I should mention, is that we have in the training set, we have 50,000 drugs and around 100 organisms. But since we have only 60,000 examples, you can basically figure out that on an average, each drug is tried for at max once or maximum twice for different model organisms that are there, or different viral proteins that we are interested in. So given this information, what we show here is that when you use the vector representation and use the different type of traditional machine learning models, what are your quantitative mean absolute errors, mean squared errors, the correlation with the true p-Campbell value, and then the R-square, or the power of your models. And you can see that all of these techniques, whether it be the end-to-end deep learning model or it be the traditional machine learning model, they are very comparable in their performances. So that's, so it's a good thing in the sense that all these models are very comparable in their performance and have high, in general, they are qualitatively, they have good performance. Here I also, show here a GAN plus CNN model. What is the GAN? GAN is basically a graph attention network. So for the SMILES, RDKit also allows you to consider the SMILES not necessarily as a linear sequence, but as the drug representation can be considered as a graph also. So the GAN is a graph attention network, so which takes as input a graph-based representation of the drug, and then we have the CNN, the primary sequence representation of the protein when we are building the model. So given all of these different models that we have, we also show here that on the test samples, like here we fit the purple curve is basically the fit of the true predictions that are there, and the spline basically fits the true prediction value here, and how all the different models are fitting the true prediction values on our test set. And we can see that all of these models can clearly follow the spline pretty well, and that's why the correlation value, power value in our model were pretty high.
So you've been trying to fit the activity, right? That's the
exactly one. Exactly. This is the activity, and here this is just a representation of it. But if you really want to understand it, the true prediction is this dot here, and along that dot on the same y-axis, you have the predictions of the different models. So the farther it is from the true prediction, the more error you will have in your activity value. The activity value is represented here by the standard p-Campbell value that we are using. But what is good is that the underlying fit, if you fit a spline to this set of test samples fitting, you can see that your models can clearly follow the spline of the true predictions pretty well. Okay. So coming to the SARS coronavirus, what we did here and what I'm showing you here is that for each of these 3 proteins which are of interest to us, as I mentioned earlier, the PL-PRO, that is the NSP3 protein, the 3CL, and the spike protein, we used all of these 7 methods that I have mentioned here and got a rank list of drugs for each of these methods. Then once we have for each of these viral proteins, and then once I have got these rank list of drugs, I did a rank aggregation, basically using average rank. And then here what we are highlighting are in this big box here are the set of drugs which appear to be the top ranked drug, showing to be the most effective against all of these viruses. The plus here represents those drugs which are coming up in the list of top 15 drugs for and are common to all the 3 viral proteins here. And then the star represents those drugs which are basically appearing as in the top 15 list of 2 out of the 3. And then individually for some of these viral proteins, we have other drugs which are coming in the list of the top 15 specifically for these viral proteins. So many of the drugs that Ehsan showed in one of his starting slides, like lopinavir, ritonavir, are appearing, consistently appearing for all the 3 viral proteins. Then also things like hydroxychloroquine and remdesivir appear for the spike protein as well as for the 3CL protein. What is important to highlight here is that many of these drugs are not necessarily antivirals. They are anti-cancer drugs, they can be antifungal drugs, antibiotic and malarial drugs, which can be used for the particular virus to target against the viral proteins. So this is where the true power of drug repurposing lies. Like, you have identified drugs which are being used in other areas and for other diseases, but they are showing that they can be effective against these viral proteins and can be used to target the SARS coronavirus. So this is just a quantitative validation, but now coming to more qualitative validation, or how can we really be sure that are they really useful, or is our model wrong? Okay.
So you're saying that there are at least 5, if you go back, there are at least 5 anti-cancer drugs which are potentially, because that's the pink color, right? Yes. Anti-cancer is the sort of pinkish color. So there are 5 of them which are potentially useful for SARS, for COVID.
Yes, and they have like high, they can be effective against at least 3 of the primary or the main proteases of the SARS coronavirus.
And have they been mentioned in the literature at all?
So let me come to that. Thanks a lot for pointing me to that. Exactly. So here we are showing you validation through exactly as Sanjay mentioned, either through literature or through clinical trials. So what you see here is that, let me first explain the first part of the figure, which is this part. You see here that this heat map represents clinical trials which are going on for variety of the coronavirus families, viruses belonging to the coronavirus families. And you can see majority of the drugs that we mentioned in the previous slide, they are present in here for at some stage or the other in their clinical trials. But what I also highlight is that these anti-cancer drugs that I mentioned, they have been also shown in literature this year only recently through either drug repurposing or molecular docking, that they can be effective against the SARS coronavirus also. Though here there are no clinical trials going on for these anti-cancer drugs, or even for some of these other drugs like I think this is a malarial or antifungal drug, but there is evidence in literature. Yes, there is evidence in literature through either techniques like ours, like drug repurposing or molecular docking, that people have shown that these drugs can be effective against viral proteins produced by the SARS coronavirus. So this is one way of validation. Let me take it one step further. So we also show you molecular docking results that people have published or are submitting to various platforms. And here they are showing you, here we are showing you 4 drugs, ritonavir, lopinavir, remdesivir, hydroxychloroquine. So these are the drugs, and these are the amino acids with which they are interacting. So this kind of interaction should be very much possible by our model also to capture, because our model also has this primary sequence of the amino acid as well of the protein, as well as the SMILES representation of the drug. So it should be able to capture that these set of interactions which are happening are driving the affinity scores or the activity scores. So when I say interaction, this interaction is nothing but a bond. It can be a covalent bond, it can be polar bond, it can be ionic bond. So different kind of bonds that this drug forms, and by forming these bonds, it inhibits the activity of this particular or the functionality of this particular viral protein. So here people have shown examples for the spike protein. What in the same example, what they showed is that drugs such as hydroxychloroquine and remdesivir have very low binding affinity. So to read this particular figure or this particular bar plot, what you have to understand is that you want your affinity scores to be absolute affinity scores to be as high as possible, or the negative affinity scores to be as low as possible. So drugs such as hydroxychloroquine and remdesivir, they have relatively low affinity scores in comparison to antiviral drugs such as lopinavir and ritonavir, which basically suggests that it kind of makes sense that all these anecdotal evidence or people, presidents of countries making a remarkable statement without even understanding that whether these drugs can be effective or not, have to be taken with caution. So like even like remdesivir is not as effective as being projected. Based on like computational models, you can already see that the affinity scores are lower. What is also interesting is that some of these drugs like mitoxytroin, which is an antibiotic, which is also in our list for 3CL Pro, has a high binding affinity for the 3CL Pro. It's published in literature, and here we also showcase what are the different protein amino acid with which it interacts for the 3CL Pro. Finally, what we also did is that we performed a docking experiment. So what we did is that for the list of the drugs that were common to all the 3 viral proteins of interest to us, we found that there is a tool called COVID-19 Glamorous AI, which allows you to dock and your particular drug by just providing the SMILES representation of your drug against the 3CL Pro. And they have a baseline score. So what they know is that the N3 ligand forms a covalent inhibitor for your 3CL Pro viral protein of the SARS coronavirus. So when it forms a covalent inhibitor, it's a very strong bond, and it can basically completely inhibit the functionality of that. And it's unpublished right now, but I mean, it's put in archive and it's under peer review. But what is interesting is that they have a lowest energy or a binding affinity score of like -7.9. So what we are looking for are drugs which can have scores in the similar range or lower range. So interestingly, majority of the anti-cancer or majority of the antiviral drugs, they have scores which are very close to -7.9. And what is more interesting is that these 3 anti-cancer drugs, the regorafenib and the sorafenib and raloxifene, all of them have much lower binding affinity score in comparison to the N3 ligand. So they can be really good candidates to investigate. Again, drugs such as the antibiotics like amonincin and mitoxytroin are also good candidates. But hydroxychloroquine, which is again overhyped, might not necessarily be as effective for the 3CL Pro protein protease as it is being touted. So, I mean, before making very strong assumptions or very strong conclusions, what I would definitely like to say is that our models are based on simplistic representations of the drug and the proteins, and they can definitely be enhanced. So all the results that we are showing should be taken with a little bit of caution, and not we cannot draw too many conclusions out of them. So what can we do? Basically, we can make use of additional information, and that's where help of other people within QCRI and outside from the community can come, and they can help us in the sense that we can use more information about the drugs, like the molecular weight or the mechanism of action. So mechanism of action is available in the form of summary for majority of the drugs. So someone from an NLP background or natural language background who is an expert in basically converting text to vector representation can be of aid there. Then we can also use more information about the viral proteins, like their biological processes that they're involved in, or their different characteristics, including like the molecular weight, grav index, aliphatic index, properties, solubility properties, many properties of the viral protein we can include in our vector representation or even in the end-to-end deep learning models to enhance them further. Then, the one solution that Ehsan presented, we can also take into account. So the function that we are estimating is basically a function of drug and the viral protein. But we have not considered a very important factor which is there and has to be used when we are making our model, which is the human cell or the human host. The function should ideally be a function of a drug, viral protein, and the human host. And one way to incorporate that information is that there is a plethora of
Now the house. The recording's still on though.
Hello.
Oh, okay, that's okay. I think maybe. Yeah, keep continuing, Raghvendra. That's fine.
It's about to finish.
I'm here.
Yeah.
No, because when I claimed the host, it muted them all, so I had to unmute them.
That's okay. Yes.
So for incorporating this information, we can use information such as like the drug human protein target, the drug viral protein target, human protein viral protein interaction, like information about the ACE2 receptor and the TMPRS, which is interacting with the spike protein, and the human protein interaction. So all this information can be included together in a knowledge graph. And one of our colleagues, along with Dr. Sanjay, like Dr. Stefano, we are trying to use the knowledge graph based representation and try to do a graph convolution neural network based approach to identify a prioritized list of drugs for the viral proteins of interest. So these kind of approaches can also be used. However, so these are some of the future work that we are planning. All of this research is unpublished. So I would again reiterate the importance that take whatever we are presenting with a word of caution and not draw large conclusions out of this. But what we can showcase here is that AI can definitely aid in the exploratory analysis part that Ehsan explained, that we can use AI to reduce the search space for the drug candidates of interest through these machine learning models in combination with molecular docking simulations, and show that we can get a list of a highly precise set of drugs which can be effective against the SARS coronavirus.
Good,
now
thank you.
I will
Thanks.
conclude and open the floor to questions.
So so there are a few questions. One was, I guess this one is quite technical. Is the predicted compound bind to the active site or some other allosteric sites?
So that's a good question. I mean, when I take into account, I take the protein sequence. I just look at the fragment, which has been shown in PubChem or in Chembl with which it interacts. Basically, if they mention that it interacts with the NSP3 or NSP2 or the 3CL, I take only that part of the full protein sequence, because as Ehsan mentioned, the coronavirus is a huge virus. It has like 30,000 genome. The size of the genome is 30K, and the viral protein the full protein size is 7,000 amino acids. So we only focus and cut on those particular proteases which are basically involved with the various known functional proteases like the NSP3 or the 3CL Pro. So yeah, we only looked at some fragments, and in these fragments you have the active sites which are there.
So another question was more about feature engineering. Is there any feature engineering involved or just dumping the raw data as input? I guess you answered that, but perhaps you want to say a few more words about that.
So feature engineering is required for the machine learning models, the traditional machine learning models, but not necessarily for the end-to-end deep learning model. For the end-to-end deep learning model, there is just dumping of the data, but for the traditional machine learning model, the generation of the latent space for the SMILES as well as for the protein sequence is feature engineering in a way.
Maybe Ehsan, you could answer. It's a more high-level question. In Qatar, are drug testing, such as clinical trials, also managed by an FDA-like institute? You know, how?
Like honestly speaking, I have no idea about it, but I can speculate that this might not be going on in Qatar.
I see. Yes.
Yeah.
Great, great. Thank you very much. I think it was very, very informative, and I hope everybody enjoyed the lecture, and hope to see you on Thursday. Thank you very much. Thanks, Raghvendra.